Hi everyone, and welcome to the webinar. My name is Tammy Ben-David, and I'm your host for today. In today's webinar, we're going to be discussing the topic, the perfect particle, why size matters. Now, particle size is a really fascinating topic because it is one of the first physical properties to be considered in an API, in an active pharmaceutical ingredient. Now, it's really important for you as a pharmaceutical company to make sure that your API supplier is able to customize the particle size distribution, or PSD as we like to call it, according to your exact specifications. So the webinar is going to be 30 minutes long. In the first 20 minutes, we're going to be discussing what particle size means, how particles are measured, why APIs are milled, and what top technologies are used for milling. And after that, we're going to open up the floor for questions. So if you have any questions while our speaker is talking, please feel free to pop it into the message box at the bottom of the screen. You'll see two little speech bubbles. So just put all your messages in there, all your questions in there, and we'll try to get to as many as we can at the end. So now, introducing our speakers. Our speakers today are top R&D experts from Teva API. The first one is Zohar de Valença, who is Senior Manager in Manufacturing Sciences and Technology. His expertise is in developing powder grinding processes, mechanical processes, and particle flow processes. Welcome, Zohar. Thanks for joining us. We also have Enrico Bettattini, who will be answering your questions at the end. Enrico has been at Teva API for 15 years, and his specialty is in powder technology and micronization. Thanks for joining us today, Enrico. So I think we're ready to begin now. So I'm going to give the floor over to Zohar.
Thank you, Tammy. It's great to be here. So when we start, first of all, we should think, how do we measure particle size? What is particle size? We can see that particles come in many shapes and forms. Some of them are easy to characterize and some of them are quite difficult, depending on the morphology and the shape of the particles. Because of that, what we should do is we convert each particle into a sphere. This is done theoretically. We don't change the particle itself, but we do represent each particle with a three-dimensional sphere. And the reason we use sphere is because it can be measured with only one dimension, which is the diameter. So if you imagine a particle represented by a sphere, we do not need to use three different dimensions like length, x, y, or z. We can use only one dimension, which is the diameter of the sphere. The conversion itself is done either by volume, by mass, or we can use every other properties, depending on what we are trying to achieve and what we are trying to measure. But keep in mind that the conversion itself is only theoretical, and the sphere that we are measuring is equal to the particle it represents, again, either by volume or by mass, for example, depending on the property we want to evaluate. Now, this is for a single particle. We need to characterize the sphere that represents it. And as we know, bulk is not comprised of single particle, but it is a statistical population. Usually it behaves in a normal distribution. And when we measure particle size, we don't measure a single particle, but the entire population. The output from measuring is statistical data, which gives us the particle size distribution, PSD. At this point, we want to discuss what are the small particles, what are the mid-sized particles, and what are the large particles in the bulk. And for this purpose, we use the three dimensions. We use the D10 or D01, as you can see on the screen. We use the D50 and the D90. The idea here is to see clearly what are the large particles in the bulk, which is represented by the D90, what are the average size particles, which are usually the main population of the particles. This is the D50. And what are the smaller particles in the bulk, which are represented by the D10. So once we have those values, it's easy to compare samples, it's easy to set specifications, it's easy to have a clear language regarding the bulk that we are dealing with. When we know how big are the smaller particles, how big are the big particles, and how big are the average particles. This is the general idea. Keep in mind that the distribution is smooth. So we can have other dimensions measured also. We can have the D10, meaning the largest, the D100, sorry, the largest particle in the bulk, or any other dimension that we are interested in. But usually having those three dimensions are quite enough. Now, it's very important to understand the measuring method. Our result is as good as our method. Usually, when we choose a measuring technology, there are a few things that we need to consider. The first one is how accurate is the method. For example, we analyze the particles in doesn't matter in any method that we use, and we compare it to microscope pictures just to make sure that it makes sense, that the particles that we measure are really there and the results are accurate and represent our sample correctly. Another thing that we need to evaluate is how repeatable are the results. For example, I will take one sample and measure it more than once, usually twice or three times, and I expect to receive results who are quite close to one another within a margin of error. So when we choose a measuring method, we need to make sure that the method is both accurate and repeatable. Furthermore, we make sure that the method is repeatable between labs, between different labs, meaning that the same analysis conducted in two different labs should yield quite close results. So we have three main methods for analyzing particle size. The first one and the most common one is laser diffraction. We use a device that lights a laser beam through the suspension of our sample. And by the way, the light refracts from the particles, there is a way to determine the size of the particles. This method performs the conversion to spheres according to volume. This is something that is derived by this technology, and this is why, because this technology is the most common one, this is why we usually, when we speak about particle size, we speak about particle size by volume. The other method is by sifters. We use sifters with known hole size, and we introduce the sample. We measure the weight of the sample, and we measure how much weight was left on each sifter, and by that we can determine the particle size according to how smaller the particles were to transfer through the holes in the sifter. So this is a second method that we use. It's less common than the laser diffraction, and it's pretty old, but it's still a very effective and straightforward way to measure particle size. The third type that we use is image analysis. We just take a small sample from our batch, and we take microscope pictures of this sample and measure particles quite by hand using measuring software. And this way we can determine the particle size according to a picture. Another two properties that are very important to understand are the bulk and tap density. For powders, they are pretty special because we have two types of densities, unlike liquids or solids, for example. We have bulk density, which is the density of the powder at rest. If you can look when the powder is at rest, we can see that there are some small particles, some large particles, and a lot of air between them. This gives us a certain amount of product per volume, which is the bulk density. We measure that by pouring some sample inside a beaker and measuring the height of the powder in the beaker. This gives us the volume of the sample, and of course the sample was weighed in beforehand. So this gives us the bulk density. Another property that is important is the tap density. After we tap or compress the sample, we can understand that the small particles realign and fill the gaps between the large particles, which means that the volume of the entire bulk reduces. This increases the density. And when we do that in the lab, we do that by using a tapping device, which taps by a predetermined program. And that way we can evaluate how much did the product compress, and we have another value, which is the tap density. So why we mill the API? The first of all, the most important property that we can control by controlling the particle size is the dissolution profile. We know that smaller particles tend to dissolve much faster than large particles, and faster is not always what we want, but it depends on the desired dissolution profile. By controlling the particle size of the API, we can really directly control the dissolution profile and have the desired results for the final dosage form. Another thing that we can control by particle size is the flowability and handling. Usually, large particles tend to flow much better and be less sticky, and vice versa. The small particles tend to flow much less better than large particles, and it depends on what we are trying to achieve. Another thing is the compatibility with the formulation. It depends if our formulation is whether it's tablets or capsules or maybe inhalation product. It really depends, and every other formulation requires different particle size in general. And last but not least, we need to make sure that our API is smooth flowing powder without any aggregates. Some APIs tend to form aggregates and lumps in the bulk. So we mill them in order to achieve a smooth and free flowing powder. Now, we have many different milling technologies in Teva API. Here you can see in this graph the desired range of D90 and the technology that helps us to achieve such a D90. So this is a general range. Of course, ranges may be different for different products, but this is in general how we adjust our processes, how we initially select the milling technologies that are going to be used. Here in this presentation, we are going to discuss three most common ones, which are the cone mill, the F10, and the Aerojet Micronizer. But other technologies are also available in Teva API. So the first one is the cone mill, is the least aggressive one. You can see that it is meant for large D90, ranging from above 100 and up to 1,000 microns, really large particles. Here, what we have is a conical screen with a rotating impeller in the middle. And the feed is introduced from the middle. This is the dry product before milling. And once the impeller is rotated against the screen, the particles are first impacted by the impeller, and after that they are sheared by the screen. So below the screen are the final API, the final particles after milling. What we control here is the screen type and size, the size of the holes in the screen, and the impeller speed. These are microscope pictures of a product that was milled in cone mill. You can see that there are still large particles. There are still particles that weren't completely milled and weren't completely broken. We can still see large particles, and also we have some fine particles, some dust around them. So this is a very coarse milling with large particles as a result. The next technology is an F10. This is a mill that introduces two millings in one pass. The first one, it is a milling chamber, which is made of two milling chambers. The top chamber is very closely similar to cone mill, when we have a conical screen and then a rotating impeller in the middle. Also here, the feed is from the middle, from the top. The first initial milling is done on the top chamber. The only difference here between cone mill and F10 is the additional impeller on the bottom. So on the bottom we have another rotating impeller that provides a secondary impact for the particles, which tends to break them into even finer dust. So here the milling is much more aggressive because of the lower chamber with another impeller. One more feature that this mill has, because we are using high RPM and we introduce a lot of energy, it tends to heat up. So this mill has a cooling jacket in order to keep our product cool and not to affect any chemical properties. So this is a microscope picture, is an example for a material that was milled in F10. Here we can see much less particles, only lucky few that they didn't break all the way. Most of them are broken and milled, only few large particles and a lot of dust around them. The next technology is a Aerojet Micronizer. The micronizer uses only compressed air without any moving parts or mechanical shear. Here we introduce the particles using a strong stream of jet. Usually it's air, sometimes it's nitrogen. We accelerate the particles into the micronizer chamber, which is round. The particles accelerate and move in a circular motion. And there are several other jets of air on the perimeter of the milling chamber. The idea here is to get particles to collide with one another. So we have particles moving with high energy, with high velocity, colliding with one another, and the entire energy of the collision makes the particle break. Both particles break into fine dust. Here, another effect is the escape. Escaping from the chamber is done from the middle, and because of the way that chamber is built, only small particles can flow from the perimeter into the middle, and that way we get some sort of segregation effect, where only small particles can escape the milling chamber, and others keep rotating and keep colliding with the other particles until they are small enough and are good for us to collect. So here the small particles after mechanization are exiting the micronizer from the middle. And here you can see microscope pictures of a product that was micronized using a Aerojet mill. And as you can see, there are no particles left at all. Everything is dust. Everything was totally micronized. And this is why we use this when we need very small particles in the API. So Tammy, back to you.
Thanks so much for your presentation, Zohar. I definitely learned a few new things today, so I'm sure our audience has too. So while Zohar was speaking, we received a whole bunch of questions from you guys in the audience. And so I want to touch on a few of those questions today. And Enrico is going to answer them. So Enrico, are you ready?
Yes, thanks. Thanks, Tammy.
Okay, so the first question is as follows. We were asked, from my understanding, the same material or sample could give different PSD results on the base of the chosen analytical method. Is that right?
Oh, yeah, yeah. The question, yes. Okay, start from the analytical point. That was the beginning of the Zohar nice presentation, and give me the chance to emphasize some details. we mainly focus on laser scattering analysis. And result, I have to say, by this method are not completely objective. They are slightly subjective, affected by the method in use. This is generally negligible, but not in the case of a respiratory application or with APIs that are characterized by a low solubility or a low permeability. The main difference in laser scattering analysis concerns if the method itself used to analyze the API as a dry powder or as a dispersion in a specific media. It is a statistical analysis, so analytical results are always related to the mathematical algorithm that has been chosen. And they are also affected slightly by the equipment model and by the sample preparation. So you must take care of the method itself.
Great, thank you. The next question is, you differentiated the milling, or Zohar differentiated the milling and micronizing equipment on the basis of the PSD to be achieved. Is that the only feature for the choice, or are there others?
No, there are others, yeah. I have to say to the listener that common physical specifications are focused on the most known particle size distribution. But many critical quality attributes of the power have to be considered for achieving the proper bioavailability for the patients and for a good formulation. So it's not only milling or micronized, not only spray drying or lyophilize. This is the challenging point, to choose the proper powder and engineering approach, the details of the activity and the operative parameters.
Okay, great. Here's another one. Are there side effects during operative particle engineering of APIs? Any side effects?
Okay, it's like life. Okay, yeah, yes, the side effects may occur. Yeah, powder engineering is mainly a mechanical activity, so APIs may undergo to mechanical stress and also to heating. So they may be affected by chemical purity decrease or decrease of their crystallinity structure and also of their stability over the time. So for this reason, another key point is control during these activities to control the amorphous content in parallel with the particle size distribution.
Perfect. Thank you. We have, Enrico, time for one more question. So that question is, what are the latest techniques and analytical trends with particle engineering?
Yes, you have to consider that modern drugs are more specific, more selective for the patient. And so usually APIs are more complex right now and more sensitive. So the new technical approaches are focused to be more specific and more delicate as well, capable to reach more selective specification ranges and less aggressive, as I explained in the previous answer. So micronizer or mills for specific purposes have been developed in this decade, and that includes high-tech construction geometry and some new modern material in use. From an analytical point of view, there are new techniques that focuses especially on morphology of crystals or rheology of the bulk powders. And they are based on new, imaging software and electronic sensor. So there has been developing also from an analytical point of view. We didn't spend a lot of time discussing this up-to-date technology in this webinar, but I think for sure it will be a future meeting between all of us.
Definitely. Thank you so much, Enrico. I appreciate your time. So unfortunately, that's all we have time for today. Don't worry if we didn't get to your question. I promise you that we will. Our experts will take all the questions that we didn't answer and send you a reply in the next couple of days. So you can expect that into your email inbox. And we really enjoyed today's session, and we hope you did too. If you'd like any more information about our products or services here at Teva API, feel free, please, to reach out to us. You can see here our email address and the website. On the website, too, there's great content, more webinars, previous webinars that we've done, podcasts, and lots of blogs and industry news. So really, thank you for joining us today, and we will see you at the next Teva API webinar. Bye.